All right, so we're back. This will be lecture part 2, but we gotta just keep on right on rolling. All right, so we drew an amino acid. And now we're going to draw another amino acid. What I'm trying to demonstrate here is when you're trying to link biomolecules, look for how you can make a molecule of water like this. Here's another amino acid. We've got nitrogen with its hydrogens, the carbon, and blah. Now, when we're going to make and start linking together polymers, what you look for is how can you make a molecule of water, or what's called a condensation reaction, in which we actually are pulling out a molecule of water, or also known as a dehydration reaction. I think the book is called it a dehydration synthesis. So in order to link these two, we're going to take the hydrogen off that guy, we're going to take the OH group off this guy, make a molecule of water, and bing, generate a covalent bond. And that is how we start linking pretty much all of our biomolecules. All of our monomers get linked through these dehydration reactions where you're pulling a molecule of water out. Now, the opposite holds. If you want to break it, we're going to break it by adding a molecule of water and splitting this bond. All right, so that's the term I want to throw in there. Now, what we want to do is build up kind of a chart in which we've got the monomers and the polymers that they make. And we've got a set of 4 basic macromolecules, and they are this. We've got, this just makes more sense. I'm putting the polymer over here and we'll put the monomer over there. We have proteins, and proteins can go by a couple of terms. They can also be called peptides if they're not very big, but proteins is typically how I refer to them. Proteins are made up of amino acids. We've got carbohydrates that are made up of sugars. Another term for carbohydrate is called a polysaccharide. That probably would be a better term to throw up here. So a polysaccharide. Not very creative because the monomer is called a monosaccharide. All right, so then that's number 1, number 2. Number 3 on our list here is nucleotides and nucleic acids. So the polymer is referred to as a nucleic acid. This is DNA and RNA. And the monomers for these guys are what are known as nucleotides. Last but not least, we've got the lipid class or fat class. And for our purposes, we'll say the polymer is a triglyceride, and the monomer for that guy is something called a fatty acid. So, what we're going to do is go through some examples of these different classes of biomolecules and look at some of their characteristics and some of their interactions. So let's do that. All right. Let's start. We're going to do it's like organization-wise, I'm probably going to lose points. We're going to start with carbohydrates, then go to fatty acids, and then do proteins, and then last nucleic acids. Just because that's the way it's done, man. Don't give me a hard time. All right. So the first polymer that we're going to talk about is the carbohydrate or polysaccharide class. And how's the red reading? Can you read the red fairly well? Well enough. All right. Polysaccharides. These are made of oh, excuse me. Oh my goodness. They're made up of sugar molecules, and sugar molecules can come in a variety of different carbon numbers. We have what are a class of sugar molecules, and these are things that you know of, like the 6-carbon sugar glucose is a sugar. There's a 5-carbon molecule that is pentose. And one of the hallmarks, a couple of things, is that the sugar molecule can actually exist in a couple of different isomers or shapes. And glucose is a 6-carbon sugar that looks a little bit like this. In that we're going to draw it as a line 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5. Oh, one more. And it's got lots of OH groups coming off of it. And at this carbon, it's got a double bond oxygen. All right. Now, when we look at this, first off, this molecule can swing around and form a cyclized structure in which this carbon is going to interact with this oxygen. And we end up with a hexagonal structure. You don't need to draw the structure. I'm not going to be worried about it that way. Save that for when you decide to go into organic chemistry. But the main takeaway point, when we look at a sugar molecule, it's got a lot of OH groups. And remember, oxygen being electronegative is going to pull that electron away, and we have the ability to form lots of hydrogen bonds. And that's an important thing that carbohydrates can do, is they are great at forming lots of hydrogen bonds. We're going to start linking together glucose, as an example, and we can make a glucose polymer. Now, the exact linkage you don't need to even worry about. It's going to look something like this. And I haven't drawn all my structures. You don't care. In that we have a link between the carbon like that. And we start linking them this way. So we're linking this carbon with this carbon. And we start making a great big structure. Glucose polymers, you're familiar with several of them. A standard glucose polymer is known as cellulose, also known as wood. Another polymer that you may be snacking on for lunch, and that is starch. So these are glucose polymers, and they can be very, very large. They can have tens of thousands, hundreds of thousands of individual glucose molecules. So you can take starch and break it apart and generate a lot of glucose out of it. And they are very polar. They dissolve really well in water. And they're going to function primarily as an energy storage system, but they also function to hold a lot of water as well. So we have a variety of different kinds of sugar forms in our bodies, and bacteria do. We use glucose as a primary form of energy, breaking the carbons apart, and we're going to go through respiration, cellular respiration later. We'll talk about that. And we also can store energy in the forms of glucose polymers. We store energy in the form of a molecule called glycogen. And bacteria have a lot of polysaccharide covering their outer walls, kind of like a candy coating. And so this is one of our major biomolecules. Thinking about my food class, one of my reasons that I love carbohydrate polymers so much is that they make things thick. So cornstarch as a thickener, or flour as a thickener. This is what puts the pud in pudding. This is what makes pudding thick. All right, so that was the polysaccharide class. Their number one claim to fame, hydrogen bonding. Now, they also are great energy storage, but all these guys are. Our next class is the lipid class. Lipids, fats, fatty acids. These are typically not water soluble, and they pretty much contain just hydrogen and carbon, a little bit of oxygen, known as hydrocarbons. So a fatty acid will typically, that's the monomer, a fatty acid will typically have 18 carbons, and it will have a carboxylic acid tail. So it's got this teeny bit of polarity to it, but it is such a gigantic amount of nonpolar carbon that it's not water soluble. So an example of a fatty acid would look like this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 16, 17, 18. 18 carbons. And on this back carbon here, we've got the carboxylic acid structure. And then the rest of these carbons, we're going to go ahead and saturate this and just put hydrogens here. I'm not going to do this. You can do that at home. Hydrogens everywhere. This makes a very non-polar molecule. Now, what we're doing here, all right, maybe I will do it, is we're making what is known as a saturated fatty acid that is saturated with hydrogens. And it is pretty much just a straight chain. A lot of energy in here because each one of these carbons can be broken off and blown off as CO2. Each one of those carbon bonds has got energy in it that we can use. And just if you don't know, oil burns really, really well. This is gasoline for all intents and purposes. So these are fats. Now, one of the things that is interesting is that we can actually have double bonds here, and a double bond will cause the removal of the hydrogens on either side of that double bond, and this puts a little kink into the fatty acid. So this is, as we've drawn it, is what's known as a free fatty acid. And if you've ever heard of omega-3 fatty acids, you're talking about an 18 carbon fatty acid with a double bond at carbon 1, 2, 3 right there. So it's not all that complicated. And these are common in lots of different fats. When you're looking at olive oil, you're looking at a mix of free fatty acids and then complex fatty acids. Blubber, lard, bacteria have fatty acids. All organisms do. And typically, we have free fatty acids, or we can begin to link them together. This is kind of our polymer version of a fatty acid polymer, and that's what's referred to as a triglyceride. And it looks like this. So we've got a glycerin backbone. Glycerin is a 3-carbon molecule. Each carbon has got an OH group coming off of it. So it's known as a sugar alcohol. And glycerin is fairly viscous, fairly thick, and also vaguely sweet. It's actually found as a humidifying agent, lots of baked goods. It's also found in cosmetics, all kinds of stuff. So what we're going to do is begin linking fatty acids to this molecule. And we do it in the following way. We have the fatty acid, and I'm going to draw it, rather than drawing all the carbons, we'll just draw it like this. There's the terminal carbon that is the acid part, and then the long carbon tail. And what we're going to do is link this together through a dehydration reaction. We're going to condense a molecule of water. So let's look to see how can we make a molecule of water out of this, H2O. We're going to take 2 hydrogens, 1 oxygen. So we squirt out a molecule of water, and we're going to make a carbon-oxygen link. That's how we begin doing this. Now, we can put 1 fatty acid tail on here and make a monoglyceride. We can put 2 on, make a diglyceride. Put 3 on, you made a triglyceride. As you get older and you start doing having your blood work done, they give you your cholesterol number, and they give you your triglyceride number. Triglycerides are one of the ways that we have fatty acids being maintained in the body. Within the lipid class, we've got a couple of interesting lipid-type molecules that are worth talking about. First off is another really cool molecule that we'll be talking about later, in that we're going to put a second fatty acid on here. And this is a diglyceride now. On this carbon, though, we're going to put a phosphate group on here. So the phosphate group, PO4, is carrying a gigantic negative charge. And that makes for a very interesting molecule because we've got this big non-polar stretch, and then we have this charged head. And this is known as a phospholipid, and it's actually the major component in cell membranes. We'll talk more about cell membranes in the next chapter. We start talking a little bit about cell biology, but just so you know. So the phosphate gives that big negative charge. That end is soluble in water, and the fatty acid end is going to be soluble in fats. Let's see. So another member of the lipid type class or nonpolar class, not based on the fatty acids, but worth mentioning, is various kinds of sterols. Starting out with the molecule cholesterol, which, if you ever have to memorize the structure of cholesterol, I feel sorry for you. It is a heterocyclic molecule. Excuse me. It is the base, though, for all kinds of different steroids. So steroids are based off of cholesterol. Just as an interesting point, if you or people you know have high cholesterol, and they can go on a low-fat diet, which really doesn't work very well, we biosynthesize, we make our own cholesterol. And so even though you're eating a zero cholesterol diet, your cholesterol can still kind of stay very high. And that's because the biosynthesis of it is not being regulated properly. Cholesterol biosynthesis, though, can't be shut down because it is essential for making steroids. Things like estrogen, testosterone, corticosteroids. People talk about prednisone and dexamethasone. Prednisone's been is a good anti-inflammatory. These are steroid hormones that are based on the cholesterol molecule. All right. Next, we've got perhaps the most important biomolecule, and that is the protein class. And the protein class, I'm going to shift back to my black marker and see how that does. Uh, proteins. Proteins, as we said, are made up of amino acids. And that's what we were drawing when we were looking at our very first dehydration synthesis example, is amino acid. An amino acid has an amine group and then a central carbon and the carboxylic acid. Now on this central carbon, there are one of 20 different side chains known as R groups. And so we generally just write this as R. Now, if you have friends that are taking biochemistry this semester, they're having to memorize all 20 R groups. We don't got to do that. But these R groups have different characteristics. And that's what's actually really cool about proteins, is that they're all based on the amino acid, but the R group has an effect. Now, let's go ahead and link up two amino acids, because that's actually an important process. And so we'll call this one R2. So we got R2 and R1, or R, and where's D2? Regardless, we're going to make a condensation reaction here. So pull off the OH group here, the hydrogen there, and we make a covalent bond. Important to note this is a covalent bond. And this is given the very specialized name of the peptide bond. It's just a plain old covalent bond. But one of the things to keep in mind is to break apart covalent bonds, you got to heat them up pretty high. And so this is a pretty tough bond. Now, what makes proteins really sing and dance is the fact that we've got these 20 different R groups. And we can categorize these R groups based on their properties. The R groups can be polar, and that means they can be either carrying a positive charge or a negative charge. They can be non-polar. They can have acidic characteristics, or they can have basic characteristics. They can act like weak acids or weak bases. So the 20 different R groups are carrying 20 different structures, and they have slightly different characteristics. They can be lumped together. So we've got amino acids that are polar, some that are positive, some that are negative. We have amino acids that are just flat non-polar. We have amino acids that are acidic, amino acids that are basic. And when we begin to combine these, some really cool things start to come about. And let's just sort of as a cartoon look at this. So, let's just do it sort of as a like a string of beads. So we're going to have like that. All right. So we got this string of beads here. Each line represents a peptide bond. And now let's go ahead and assign some charges to these guys. I'm breaking out some of my new colors. Let's see how that works. So we'll put a stretch of negative charges here. Can you read that as negatives? Sure, more or less. Why not? Just know I'm putting negative charges here. Now let's put a stretch of non-polar charges, non-polar R groups here. And now let's put a stretch of positive charge R groups here. All right, so what we got then is a protein that has these different R groups lined up, and when we throw them into water, some interesting things start to happen. And truthfully, this is the fundamental idea behind life, the universe, and everything. So it's some good stuff here that you're learning. First off, we can think about the different structures these things can fold up in. And we can kind of base it on some organizations and think about the bonds that fold them up. So we have what's known as the primary structure. That's abbreviated as a 1 with a degree symbol on it. So the primary structure is defined as the amino acid sequence. And it's held together by our toughest bond, the covalent bond. All right? Now, that is something that we can very easily diagram because it is simply the amino acid sequence. Held together by a covalent bond. We're not breaking that unless we set the protein on fire. Now, the next level is called, not surprisingly, the secondary structure. Now, secondary structure is defined as the R group interactions between neighboring amino acids. Who are the amino acids in your neighborhood? And neighboring is probably the guys like 4 or 5 amino acids around you. Now, just to skip ahead a bit, the bonds that are responsible for this are going to be primarily our way weaker bonds. We're going to have polar, non-polar interactions, ionic bonds, and hydrogen bonds. And remember, these are our really weak bonds. So a way of thinking about this is we got a bunch of like-charged R groups. And because they're all negative charged, they can't stand next to each other. They're negative charged, they're repelling each other, they begin to twist. And this group of amino acids is going to rotate such that we've got the charges alternating on either side. So the molecule actually begins to twist. It actually forms what's known as an alpha helix, or an alpha helical twist. Like that. So the nonpolar guys are going to start their little fat party over here. The stretch of polar amino acids, they're going to also form an alpha helical twist. But now they got their positive charges alternating. Okay? So, that's the secondary structure, the neighboring interactions. Next we have, you probably can guess it, primary, secondary. What's next? Tertiary. Tertiary interactions. So the tertiary interactions are referred to as non-neighboring interactions. And so now we're going to take this end of the protein and swing it around and interact with that end of the protein. Maybe make something that look like this. So here's the nonpolar part, and so we got the negatives and the positives, and negatives and the positives, negatives and the positives, and we can create a folded up protein. Another structure that I didn't throw in, and I'm going to mention it here just in case you see it, in the secondaries, is that in addition to forming spirals, you can also form a hydrogen bonded zigzag, where you've got hydrogen bonds that are holding this thing together. That's known as a beta pleated sheet. All right, so these are the structures. And just to complete this whole game, the tertiary structures, they're also held together by the weak bonds. That is, just to write it back down, polar, non-polar interactions, ionic bonds, and hydrogen bonds. Now, an important thing to think about is the primary structure is held together by really strong bonds. Secondary and tertiary, though, much weaker. What happens if we just barely gently heat up a protein? Say, like, bring it up to about, hot tap water. Maybe the water coming out of your water heater. 120, 130 degrees. Our water heater is pretty cranked up, probably about 140 degrees. We're not going to break the primary structure. Remember, that's only with fire. But the secondary and tertiary structure, that falls apart. So primary structure is very tough, but secondary and tertiary, you look at it wrong and it can begin to fold apart. And we have a term for a protein that has lost its primary or its secondary and tertiary structures. And it is referred to as being denatured. So a denatured protein is one that has lost its secondary and tertiary structure. A loss of secondary and tertiary structure. And protein denaturation happens actually at a very light level, a slight level, during a fever. Protein denaturation happens, of course, when you're cooking foods. Denatured proteins, sometimes they can renature, like after the bad case of the flu and having a fever, your proteins can renature fairly well. And that's part of what makes you feel so bad, is your proteins are beginning to actually denature. Other times proteins can't renature. They just won't. Once they fall apart or once they lose their structure, they can't really be folded back together again very well. This is actually a major part of how proteins work, is because of their structure. Now, not to preach, but a really important point to think about is proteins work because of their overall structure. Their overall structure is determined by their primary structure. Primary structure is dictated by their genes. So a gene, as we're about to, later we get into this, a gene is really just the instructions for what amino acid to put next to what amino acid. What is the amino acid sequence? That's what a gene is. It is the instruction set for the amino acid sequence of a protein. And if you change an amino acid in this guy, you can change its tertiary and secondary and tertiary structure. And when you do that, you can change its function. Consequently, a change in the DNA might result in a change in an amino acid, and that might result in a change in secondary and tertiary structure, and that might result in a protein that does something really weird, causes cells to begin dividing out of control, doesn't work very well, doesn't work at all. That is, in fact, the key to genetics, right there. Thank you very much. All right, let's go through one last little bit, and then we're going to call it for the day. We'll pick up a little bit on cell biology next week. So our last polymer is the nucleic acid class. And we're not going to belabor too hard this class, but this is where information is stored to create proteins. And a gene is a portion of a nucleic acid polymer. And a gene is going to have the instruction set for an amino acid sequence, for a protein sequence. One of the other things that DNA has to do, or nucleic acids have to do, is they have control sequences that say this gene should be turned on and expressing this protein now, or this gene should be turned off and not expressing this protein. So DNA is made up of nucleotides, and we got two different kind of flavors of nucleotides. We've got the base structure of a nucleotide looks like this. It's a 5-carbon sugar. So we got carbon 1, 2, 3, 4, 5. Actually, the nomenclature is like this. Sorry. This is 1, 2, 3, 4, 5. Coming off of carbon 1 is a nitrogen-containing structure that's known as a base. And I'm kind of being a bad lecturer, and that the base actually gets the straight numbers, 1, 2, 3, whatever, and then the sugar has the primes, what's known as 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And so that's how we designate the individual carbons in a nucleotide, is we have the base, and then we have the 5 carbons of the sugar. Now, on carbon 5 prime here, we have a phosphate. And remember, negative charge on that phosphate. We have a phosphate coming out. And depending if it's in the monomer form, it typically is going to have 3 phosphates. And this is referred to as a nucleotide triphosphate. Now, as I said, there's 2 different forms of this sugar. And remember, sugars have a lot of OH groups on them. We're going to just look at 2 of them. There's an OH group that is on the 3' carbon, and an OH group that's on the 2' carbon. All right. The OH group on the 3' carbon is critical because this is how we link together the next nucleotide. We're going to take a phosphate and link it to that oxygen. And this is what's known in the biz as the phosphodiester link. And we got now the OH group and the OH group here. So the 3' hydroxyl, you can't jack with that one. It's always there, the 3' OH. The 2' hydroxyl, though, you don't need it. And so we've got 2 different nucleotide forms. One that has the 2' hydroxyl, and one that has the 2' hydroxyl removed. It has been deoxygenated. And that's where the name comes from. So if the 3' hydroxyl has got to be present, the 2' hydroxyl, if it's there, is it there? I don't know. Look. If it's there, this is known as a ribonucleotide or a ribonucleic acid. If the 2' hydroxyl is missing, then this is known as a deoxyribo sugar or a deoxyribonucleotide. So we have a ribonucleotide or a deoxyribonucleotide. And then that's where the abbreviations come from. We have deoxyribonucleic acid, known as DNA, and we have just plain old ribonucleic acid, or RNA. They actually are very, very similar. You can substitute one for the other, probably no problem. Oftentimes different systems. But some organisms, viruses particularly, they actually exist as RNA molecules. In order to actually interact with cells, with you and I, they have to be converted into DNA molecules. This is the stuff of life. Now, the base here is able to interact with a base on another strand, what's known as base pairing. And the other strand is running upside down. We can look at all kinds of fancy drawings, but it's not really important to recognize that for our purposes. The main thing is the base links, or the base pairing, is that we have 4 different nucleotides: adenine, guanine, cytosine, and thymine. The thymine structure in RNA is called uracil. These are, of course, abbreviated ATGC and U. And this is how they work, is that adenines base pair with thymines to make what's known as AT base pair. Can you see that? Yeah, close enough. Guanines base pair with a cytosine. So you have GC base pairs. Now, you notice I had 2 lines and 3 lines. They actually base pair by hydrogen bonding. And GC base pairs form 3 hydrogen bonds, ATs form 2. You can cause the DNA strands to separate by boiling them up. They actually then can reanneal and reassociate, but that's not that critical. Now, DNA is double-stranded, and so DNA forms a helix, or a double helix. You can think of it like this. Imagine taking a ladder where the rungs are the base pairs and now twisting that ladder. That would be a double helix. I'll just say that, the drawings of it are not easy to do to make it look right. If you're ever drawing DNA structures, don't try to draw them as a double helix. You'll confuse yourself and you'll make your instructor cranky. But that's the way we think of double-strand DNA, as a ladder that is twisted. Now, RNA is single-stranded. But because it's single-stranded, it can actually form internal base pairs, and you can get some really interesting, cool structures with an RNA molecule. Things that look like this, where we have internal, or as we call it, intramolecular base pairing, in which you can make these little stem and loop or cloverleaf structures. So you have complementary base pairs set up in such a way that this molecule can fold back on itself and form 3-dimensional structures. And that's one of the hallmarks of RNA is able to form 3-dimensional structures within itself. Hey, that's a fair amount for today. Whoa. But we had some stuff I needed to cover. We're going to pick up on Monday and get into some basic cell biology. I'm going to post some PowerPoints and study guides for you. Things to look at over the weekend. Oh boy. And I'll be talking with you later. Hey, don't be a stranger. If you have questions, come by. If you don't want to come by, we can Zoom. I'm around. Holler. And that's all I have to say. I'm still looking for my good sign-off. If you can come up with something. I'm out of here.